So welcome everyone to our OpenSIM webinar. My name is Jennifer Hicks. I'm the OpenSIM Research and Development Manager, and I'll be serving as the moderator for the webinar today. I'm pleased to welcome our presenter today, Wendy Murray, who's joining us from Northwestern University and the Rehabilitation Institute of Chicago. Next slide. So OpenSIM is a freely available software application for visualizing musculoskeletal structures and simulating movements of humans and animals. The application includes tools for general purpose inverse dynamics, optimization to estimate muscle and joint forces, methods to create simulations from motion capture data, and then tools to analyze and visualize the results of simulations. So the first goal of our webinar series is to showcase cutting-edge research that's being performed with OpenSIM. OpenSIM is also a growing and geographically diverse community of users. So the second goal of our webinar series is to provide an easy platform for the OpenSIM community to communicate and collaborate. Next slide. So before we get started, I just have a few quick reminders about the webinar format. We'll address your questions at the end of the presentation using the Q&A panel. And if you need any additional technical help, you can consult the guide on our website. Now I'd like to introduce our speaker. Wendy Murray is an associate professor at Northwestern University with appointments in the departments of biomedical engineering, physical medicine, and rehabilitation, and physical therapy and human movement studies. She's also a research health scientist at the Edward Hines VA Medical Center. Before coming to Northwestern University, she directed a research program at the VA here in Palo Alto. Her research focuses on biomechanics of the upper limb with a focus on developing mechanically accurate musculoskeletal models, specifically applied to rehabilitation. Wendy is also an active member of the OpenSIM community. She serves on the Scientific Advisory Board of the National Center for Simulation and Rehabilitation Research, and has contributed directly to the community by publishing top quality research, sharing her models and data, and serving as an ambassador for modeling and simulation. So with that, I will pass the baton over to Wendy. Take it away.
Thanks, Jen, for the introduction, and thanks to the OpenSIM team for inviting me to give this webinar. I've gotten a few compliments from colleagues about my title. I'm guessing that it's a pretty universal experience for those of us in the field to finish a presentation at a conference and have someone in the audience say something like, well, we know all models are wrong, so can you tell me something valuable from your research? It's a little bit in jest that I chose this title, but when giving my first talk as a postdoc at the American Society of Biomechanics meeting, I did end a detailed biomechanical simulation analysis. Many of you know I studied tendon transfer, so it was one of the first analyses I did of a brachioradialis tendon transfer. And I vividly remember the first question from the audience that I was asked was, how do I know, how do you know your model is right? Well, I really do appreciate the reasoning that's behind these types of questions. When we report results from a simulation, we've studied a simplification of the system we care about. We haven't necessarily summarized something that's directly from the system, and it's really important to know that what you're telling people and what you're learning about is valuable for movement. However, I do want to bring up the point that I think questions like these can actually miss the point of the power of modeling and simulation in answering questions about human movement. And my goal today is to highlight the really valuable and unique contributions that modeling can make. Overall, understanding the actions of muscles is much more complicated than it seems. To ask a question like, what does the biceps brachii do, in some ways seems pretty pointless. From basic anatomy, we understand the mechanical actions of the biceps and all of the muscles in the human body, really, in terms of how they are attached to the skeleton and the movements they cause when they develop force. In an anatomical study, this would be evaluated by simply pulling on the muscles. And as illustrated by this classic art piece, it feels like we've known what muscles do for centuries. As simple and as basic as it seems, the musculoskeletal system and how it produces movement itself isn't that simple. For example, here I'm showing 3 muscles of the distal upper limb. The extensor carpi radialis longus, which crosses the elbow and the wrist, is considered a primary wrist extensor. The FDP, the flexor digitorum, crosses the wrist and the fingers. The FDS, another finger flexor, crosses the elbow, the wrist, and the fingers. And in none of these have I even mentioned the fact that the forearm rotates, and they may or may not contribute to that degree of freedom as well. I could give many examples of how basic approaches that we used to think about what a specific muscle does is really too simple. As scientists and engineers, one of the approaches we use is to instrument our subjects. Here I'm showing a picture from my collaborator, Kate Saul's lab, and some animations of arm movements that she collected in our collaboration describing upper limb modeling and validation. But it illustrates a very common approach in experimental science is to instrument the limb, measure joint motions, measure muscle activity, and use this type of data to interpret what the muscles are doing based on when the nervous system is activating them and how highly they're being activated. However, as the classic biomechanical problem is that there are more muscles in the limbs than really are baseline necessary. It's a redundant system. And the complication of the elegant anatomy and the complicated ways that we can move make it not so straightforward to definitely conclude what a muscle is doing and how it's contributing to the mechanics of the movement and how it's producing the movement. So for this reason, I have spent most of my career working towards developing mechanically accurate biomechanical models of the upper limb and integrating them with experimental research with a goal to better understand the mechanical actions of the upper limb and better understand human movement. In my lab, we use biomechanical modeling as a methodological tool. It advances the types of questions we can ask about human movement, and I'm going to focus in on one study today to try and illustrate how biomechanical models are not always wrong. The study I've chosen to talk about is work from a graduate student in my lab, Sarah Wallman. This work was recently published in the Journal of Biomechanics, and we used a simulation-based approach to understand gaps that exist between really valuable experimental studies characterizing the transformation between muscle force and the endpoint force produced at the thumb. So I don't have to give very much motivation to explain that thumb function is critical for daily life. Here are some basic illustrations of really fundamental tasks we do without thinking about them. And a majority of our daily activities require us to use our hands to interact with the environment. In a basic study from 1964, a clinical review demonstrated that 40% of all daily activities involve the thumb. Now, if you started to review the literature, it wouldn't take you very long when looking at studies describing biomechanical models of the thumb to jump to the conclusion that biomechanical models of the thumb are wrong. One of the reasons you might jump to this conclusion is the authors often explain to us what their model doesn't do well. I'm going to focus in on one of the premier studies in biomechanical modeling and thumb function from Francisco Valera Cuevas and my former colleagues at the VA Palo Alto, including Lisa Johansson and Joe Towles. They developed a biomechanical model of the thumb to predict the thumb tip forces produced by human subjects during maximum voluntary effort. What I'm showing here on the bottom of the screen are two really important functional hand postures. The lateral pinch posture, which you might recognize that you use to pick up a key, so it's also called key pinch posture, and opposition pinch posture, which is used for more precision grip. So they studied the ability of individuals to produce forces in these different postures under maximum voluntary contractions, and they also developed a biomechanical model of the thumb. And here I'm showing the outline of the kinematic representation of the thumb joint that was reported in that paper. So as I said, they used this model to try and predict the thumb tip forces produced in a really elegant experimental setup where individuals were required to generate forces in specific directions, and EMGs were measured via intramuscular electrodes. I'm going to jump right to the conclusions rather than getting into the details of the work. But like I said, and I'm starting out here, if you're just reviewing the literature, one of the things you might stumble across is the conclusion of this paper was that the simulations were unable to replicate the human subjects' data. The simulation results were not a little bit off, they were 4 times weaker than the human subjects. In addition, you can see from the conclusions of the paper that when they solved for the muscle activation patterns needed to generate maximum forces, they concluded that the muscle activation patterns weren't well matched to the activation patterns the individuals used when generating their forces either. So again, looking at the literature, how do you know your model is right? Well, it looks like this model is wrong. In fact, the authors themselves did detailed investigations of what parameters in the model might be leading to this inconsistency between the simulated and experimental data. And they did really complex Monte Carlo simulations where they did over 5,000 simulations to sweep through the number of parameters that and the variability from the literature that exists to see if they could do better. And this 4 times weaker conclusion is based on the results of those simulations, those Monte Carlo simulations. And based on the fact that they looked at a broad range and distribution of moment arms through these Monte Carlo simulations and a wide and broad range of force-generating parameters of the muscles, they concluded that it must be the oversimplified kinematic representation of the thumb. So what they've shown here is that they have orthogonal reference frames that are all aligned with each other, and that joints with 2 degrees of freedom, like the CMC joint, the axes of rotation intersect. So they knew this was a simplification of the joint kinematics of the thumb, and based on the detailed study they did, they concluded that it was this oversimplified kinematic representation that likely explained the inability of the model to replicate the human subject's data. So moving on to further review of the literature, this comes from my own lab with a collaboration with Craig Gaylor, a former postdoc in my lab here at RIC. We're very interested in understanding more about thumb function, and we're very aware of the work from Francisco's group. And so we started to think about another set of data that Francisco and his students and collaborators have collected. In particular, this fancy contraption that was developed through Francisco's work is a cadaveric setup where a hand is attached to a 6 degree of freedom load cell, and linear actuators can be used to power and pull on individual muscles, much like in that Rembrandt photo. You can pull on an individual muscle and record the endpoint force the muscle produced. And again, they did this in the same range of postures that we see here. My colleague and I, Craig, focused in on lateral pinch because we knew there's a lot of difficulty replicating the data. So Francisco's earlier study focused on the human subjects data, but we decided we'd look at whether the models were representing this cadaveric data. Here we could think about the individual actions of muscles because we figured if something's not happening in the coordinated actions, then there must be something wrong with the individual actions. So what we did is we tried to predict the thumb tip forces produced when individual cadaveric muscles were loaded with a known force, replicating Francisco's experimental data from Cornell. And the first author on that is Perlman. It was published in 2004. Given his results and conclusions from the 2003 paper that it was really the kinematic representation, we were lucky enough to have the collaboration that Kate Saul and I are involved in. The kinematic model of the thumb involved in our 2005 paper does replicate experimental data from Hollister and Brand. And so you can see here that these axes of rotation are clearly not orthogonal and aligned with each other, and while you can't see it here, the CMC joint, the axes of rotation don't pass through the same point. So here we've implemented a much more complicated kinematic representation of the joint, directly addressing Francisco's concern. In this case, to our surprise, the simulation results overestimated the measured forces from the cadaveric data. So again, this is pulling on an individual muscle one at a time, measuring the endpoint force and trying to predict the model, with the model the endpoint forces produced when the muscle is loaded with that. We found, given the overestimation, that the best way to match the magnitude of the endpoint force was to decrease the moment arms in the model. So again, I just want to point out, previously in human subjects data, we were dealing with data where the model was too weak. And now when we're looking at the cadaveric data, we're overestimating the measure of forces. The way to change the model to make it better match the data is to decrease the moment arms. And then we also found this interesting result that we have this really different orientation of the endpoint force vector. So here I'm showing in the sagittal plane the thumb tip. We see the proximal direction pointed back towards the body, and the palmar direction. This direction would be the direction you most likely want to produce if you were generating a lateral pinch force. And here we're seeing the results for the FPL. Here's some cadaveric data, and you can see that in the reported posture from the study, we have an endpoint force vector for the FPL, for the FPL that is misdirected and larger than the cadaveric results. And what we found was that by adjusting the thumb posture slightly and allowing there to be some variability in the individual joint postures, that we could better match the optimal posture, or better match the direction of the endpoint force. But we couldn't match the magnitude by adjusting thumb posture. We had to decrease the moment arms. So here's a summary of some of that work we did. What we did is for 4 of the extrinsic muscles, the model that I developed with Kate Holzbar and Scott Delp only included the 4 extrinsic muscles, the muscles that originate in the forearm and attach in the thumb. And so here I'm showing the FPL, the major flexor, and the EPL, the major extensor. The reported posture here is the posture reported in the Perlman 2004 cadaveric study. So you can see this looks like if the hand were here, it would be a very functional lateral pinch posture. What Craig and I did was allow there to be some variability in how strictly we enforce the limits of the joint posture of the thumb. And as we expanded the bounds of the angle of individual joints to differ from the reported posture, we found a very impressive decrease in the error in the orientation of the endpoint force. So basically, by trying to simulate the cadaveric data, proved that the biomechanical models of the thumb were wrong again. We overestimate measured forces from cadavers. We have to change the posture of the thumb, and we have to decrease the moment arms to replicate the data. One of the great things about this conclusion that biomechanical models of the thumb are wrong is that it's really replicable. So Sarah Wallman, the graduate student who worked on this project, started to do some more simulations. This time we decided to focus on the intrinsics because we're kind of a glutton for punishment over here. So again, we're trying to replicate the thumb tip forces produced when individual cadaveric muscles are loaded with a known force. But now Sarah's starting to try to add the intrinsic muscles to our model of the thumb. So here I'm showing the result from some initial simulations that Sarah did. Here again, we're in the lateral pinch posture, and I'm showing the thumb tip. This shaded red curve is the endpoint force data measured from the cadavers from the Perlman study, and then this shaded gray area represents the bounds of plus and minus 1 standard deviation in magnitude and direction. And what we're seeing here is similar to what we saw for the extrinsic muscles from Craig's study, that the simulated endpoint forces were much larger. Here we had better direction, but that was just that wasn't true across all intrinsic muscles. What's interesting is, given some of Craig's results, we went to the moment arm data. We took the premier moment arm data published in 1998 by Smuts and his colleagues. It comes from Cayenne's lab. No better place to measure moment arms than the Mayo Clinic. And these were the moment arm data that Francisco implemented in his model. We saw that even with those moment arms, and even with this kinematic representation of the thumb, based on experimental data reported by Hollister and Brand, we could not replicate the endpoint forces of individual muscles. So, as a recap, I have hopefully sufficiently convinced you that biomechanical models of the thumb are wrong. I've shown you how Francisco Valero-Cuevas and his esteemed collaborators demonstrated that forces produced by human subjects are underestimated. I've shown you how we have, in my lab, have demonstrated that the forces produced by individual muscles in cadaveric testing are overestimated. I've also highlighted some differences in the different models that have been used to generate these conclusions, including that the kinematic representation can be different across models, and that there's moment arms that are based directly from the literature and moment arms that are based from more musculoskeletal modeling techniques, and neither of those replicate the data. In addition, I've shown you that the model predictions are sensitive to the joint posture. One of the things we were most struck by that joint posture result is that we could change the joint posture of the thumb what seems fairly substantially, and it still looked like a lateral pinch posture. And finally, something I haven't measured is the peak isometric forces. So Francisco's study, they used peak isometric forces estimated from muscle volumes from cadaveric studies, and work that I've done since then with my colleagues has illustrated that muscle volumes in vivo are much, much larger than that. So there's a chance that Francisco's data or Francisco's study had peak isometric forces that were well below human subjects. So I want to stop the intro and ask you to answer the question, which set of parameters do you think are most important in simulating maximum thumb tip forces produced by human subjects? You have 30 seconds, and you should be able to see it on the screen. I really hope you all vote. It'll be interesting to me to see what the webinar attendees think should be the most relevant parameter to be concerned about. Did anybody vote? Joy, can you help me?
We're tallying the results.
Oh, okay, great. Okay, so we have a fairly large proportion of the, okay, most people are answering that most people are not answering, but 23% of the audience has chosen muscle moment arms, and 14% is choosing kinematic representation. Okay, so we'll come back to that later. All right, so despite the fact that there's a lot of evidence that biomechanical models of the thumb are not really working, and we can't really explain why, I encourage Sarah to continue to build a biomechanical model that included the intrinsic muscles of the thumb. And so the first step of her project was to define muscle paths of the intrinsic muscles. And she used a really elegant optimization strategy, which is illustrated here in this flow chart. So this optimization method for choosing muscle paths was developed in a collaborator's lab here at Northwestern, and it's referenced here in the Journal of Experimental Biology. So basically what we set out to do is optimize the origin and insertion points of the muscle tendon path so that the difference between the endpoint force estimated for a given muscle and the experimental thumb tip forces measured in the cadaver study from Perlman in 2004, that error is minimized. And so basically, we start with a muscle origin insertion. We use the partial velocity method, which is a method similar to the tendon excursion, tendon displacement method for estimating moment arms from the change in muscle tendon length relative to joint angle. We did this simultaneously for the lateral pinch posture and the opposition pinch posture because that's who we have those are the postures we have the cadaver data for. And what we get out of this part is a set of moment arms when the thumb is in lateral pinch posture and a set of moment arms when the thumb is the opposition pinch posture. We multiply that by the muscle force in the cadaver experiments. This is a known value that the experimenters applied to the muscle, and we get out lateral pinch torque. Because we're studying the intrinsic muscles, there's only 3 torques that they produce, or they only produce torques about 3 degrees of freedom. So we transform those torques through the Jacobian to get to an endpoint force, which isn't located on the distal phalanx. So we go through one more joint transformation based on the posture of the IP joint in both of these hand postures, and we get out a prediction for that muscle origin insertion of what the endpoint force produced by that muscle is. We compare that to the data, and then the iteration loops again, and we continued this optimization process until the error here stopped changing. So basically, from doing this optimization process, we produced a model that better replicated the thumb tip forces produced when individual cadaveric muscles are loaded with a known force. So here's that representation I showed you before. Here is for the ADP, the cadaveric data measured in the Perlman study. Here's what happens when we use the moment arms from the Mayo Clinic study. And here's what happens when we use the optimization process. So what you can see is, I don't have the Perlman data here, but we're centered within the plus or minus 1 standard deviation, and we're within the magnitude and orientation. And so basically, we got this type of success in most cases. So again, we were studying only the intrinsic muscles, and what we're seeing here is without the red arrows, we're just seeing the plus or minus 1 standard deviation centered where the endpoint of that experimentally measured force vector would be. So for instance, over here for the OPP, you see that we're a little bit misdirected. The optimization didn't get us in the center of the plus or minus 1 standard deviation. But overall, for both the lateral pinch posture and the opposition pinch posture, we were able to replicate the data from the Perlman study within 1 standard deviation. A new insight that we were able to draw was that the cadaveric studies describing muscle moment arms and the cadaveric studies describing muscle endpoint forces are not mechanically consistent with each other. So again, I'm showing results for lateral pinch and opposition pinch for the same 5 intrinsic muscles that I just talked about, their endpoint forces. I'm focusing in on 1 degree of freedom, CMC flexion, and I'm showing you the CMC flexion moment arms. The gray bars with the standard deviations are the experimental data from the Valero Cuevas 2003 study where they measured the maximum I'm actually saying the wrong thing here. This is experimental moment arm data. So the experimental data from the SMUT study of what the moment arms are through the 5 intrinsics. So these are from, I think, 11 cadaveric specimens. So we see a standard deviation dealing with the variability across specimens. And then the black are the model simulations that result from our optimization process, the moment arms that correspond to these endpoint forces. So what you can see, and although I'm just showing you 1 degree of freedom, what you can see here is that there's a trend for these moment arms to be much smaller than were measured experimentally. Okay, so what we concluded here was that the experimental data, which are 2 separate sets of well-done studies, but from 2 different populations of cadavers using 2 very different methodologies. Tendon displacement is like a kinematic measurement. You change length and measure changes in length and change in joint angle. And then the endpoint force is you load a muscle with a force and you measure an endpoint force. So it's pretty clear that you can't predict these endpoint forces from this kinematic data. So given that previously we had a model that over-predicted the endpoint forces, and now we've placed the endpoint forces where the cadaveric data is. it seems like we're going to further underestimate the endpoint forces when we combine all of the muscles to replicate Francisco's study. So again, what we're shooting to do here with this second part of the study is to take our augmented model, the original model from Holzbar, 2005, with the extrinsic muscles. We've added these new intrinsic muscle tendon paths, and now we're trying to use exactly the same simulation methods that Valero Cuevas used in 2003. And in particular, we're using the principle of virtual work to transform individual muscle forces into individual endpoint forces through pre-multiplication by the moment arm, and then transformation from the torques about the joints to the endpoint forces through the inverse transpose Jacobian. In addition, once we have the transformation from a given muscle's given level of force to endpoint force, we need to solve for the activations that maximize the force produced in a particular direction. And this goes with the experimental measurement. So the experimental measurement was, for instance, in one direction, Palmer, let's say it's downward in the x direction. We want to generate the maximum force possible in that direction with minimal misdirection. And the experiment was very well constrained, and in Valero Cuevas' simulation study, they used linear programming to solve for these activations to try to best replicate that data. So we're trying to maximize endpoint forces, solving for the 9 muscle activations that do that, and focusing in on a specific direction. So the end result of doing the 5,000 Monte Carlo simulations to see how sensitive these types of parameters are, the results are to these types of parameters, shows that we have accomplished something new. Our model successfully reproduced the magnitude of thumb tip forces produced by the human subjects in the 2003 Valero Cuevas study. So here, again, I'm showing the force magnitude. I don't have it labeled here, but this is lateral pinch. This is opposition pinch. Here we have the thumb showing that now we have 5 directions. We have dorsal is an upward force. Palmar is that downward force. And then we have medial and lateral and distal forces. And what we can see here, if you remember, in Francisco's original results, the Valero Cuevas original results, the model simulations were 4 times weaker than the experimental data. And just visually here, you can see that we have really improved that performance. And in general, we're across the 5,000 iterative simulations, generating an average and standard deviation that's realistically comparable to the experimental data. So a really big deal. Okay, so this is a lot of complicated stuff, and it's even hard to describe. So just to recap and make sure we're all on the same page again. Using the new muscle tendon paths generated from an optimization process for the intrinsics and a more complex joint kinematics model than was originally implemented in the Valero Cuevas 2003 study, our simulations replicated the forces produced by individual muscles in cadaveric testing. Were substantially smaller than the moment arms reported in a separate cadaveric study, but also replicated the magnitude of the forces produced by human subjects. So keep in mind, we went from a model that overestimated forces produced by individual muscles and underestimated forces produced by human subjects to a model that replicated both. So decreased the individual forces, but increased the endpoint for the combined forces. So I'm going to go back to this exact same question and ask you, has anything about the simulations we've completed led you to better understand which of these factors was most important in simulating maximum thumb tip forces? Okay, and I'm thinking they're going to tally the responses, and very quickly we'll have a result. Okay, we have more people answering, it seems, than we did last time. And we still have the majority of people choosing moment arms, and we've had more people starting to think about the kinematic representation. A lot of people have abandoned joint positions and maximum isometric forces. So we don't have enough information at this level. While we can replicate the data, both in terms of the forces produced by human subjects and the endpoint forces produced by individual muscles, we don't have enough information to be able to really conclude which matters the most. So what Sarah did at this next point was compare how joint kinematics, moment arms, and maximal muscle forces perform when we're trying to replicate the data from the Valero-Cuevas 2003 study. So what we're doing here is we are completing this study, and we're subbing in and out different moment arms, we're subbing in and out different maximum muscle forces, and we're subbing in and out the different kinematic representations of the thumb. So first I'm going to show you, for the joint kinematics, the experimentally based. So this is consistent with the Holzbar 2005 thumb model, so that's based on the experimental measurements from Brand's lab. The moment arms, we're comparing moment arms that are estimated from the model, which you'll remember for the intrinsics at least are much smaller than the literature data. And then maximal muscle forces, we're using estimates of maximal muscle forces that are consistent with volumes taken from human subjects. What you can see here is that we're getting forces produced that are on the same order as the human subjects data. That's the result I just summarized. When we sub out the moment arms from the model to the moment arms from the literature, we get what you would expect. We know we put in bigger moment arms from the literature, we get a slightly larger result. Okay. When we do those same sets of simulations, we swap out moment arms, but then we do that with the simpler kinematic representation that the Valero-Cuevas 2003 study started. You see we get a drastic decrease in the ability of the model to replicate the endpoint forces, and it's much, much weaker. So here we're seeing forces, endpoint forces during maximum activation using the same constraints decrease 3 to 5 times. So here we're replicating the results of Francisco and that study by showing that this simple kinematic representation does cause a major weakening of the muscle, of the endpoint forces coordinated actions can produce. And the last thing we did is we switched out the maximum isometric forces so that they were no longer consistent with volumes from MRI in living human subjects, but were taken directly from cadavers. What you can see here is if you compare these 4 to these 4, that again, we do get a large decrease in the endpoint forces that you can produce, expressed as a percentage of what was measured in the human subjects, but that in every case, if you compare the exact same simulations with the experimentally based versus the orthogonal and intersecting, that is the biggest difference. So our major conclusion is that in agreement with Valero-Cuevas 2003, where they postulated, but they weren't able to test this directly in their study because they tested so many other things, the kinematic representation is the critical thing for replicating forces produced by human subjects. So I'm not going to ask this question again, but I do want to bring to your attention that through concerted modeling effort, both Valero-Cuevas and our study more explicitly demonstrated that the kinematic representation. But I think the moral of the story, of course, is to never doubt Francisco Valero-Cuevas. Okay, so that's my presentation for today. It obviously was pretty detailed, and so we're not going to go into the second study, of course. What I want you to take from this, even if you're not someone who studies the upper limb or the thumb in detail and have never read about these papers, is that the simulations provided unique insights. Our simulation methods have played a critical role in understanding basic biomechanics of the thumb. If we had abandoned biomechanical modeling because, as you saw at the beginning of the talk, all the simulations were wrong, we would know a lot less about the thumb right now. Modeling provides a really valuable platform to integrate the information from different experimental sources, and you can see it helps us gain a deeper understanding of the data. It's important to realize, and this isn't a criticism of experiments, it's as there's limitations to model, there's limitations to experiments. It's not possible to measure everything we need to know. You couldn't get endpoint forces of individual muscles from human subjects, and coordinated actions that replicate human subjects would be difficult to get from cadavers. So it's a necessary problem that we have to step between different experimental techniques, and the model is a way to integrate that. We know that nothing can be measured perfectly, and here we've demonstrated how different methodologies can introduce different types of errors. An important type of error in these studies is that it's really hard to take this data, and so the studies themselves may have to be underpowered statistically because it's just so difficult to get the data. The other thing is, because these experiments are so hard to complete, the simulations here are especially useful because they can help us design smarter experiments and sort of limit the work we're doing in the lab by helping us focus on what we really need to improve our models. Another really important thing that may not be clear from the way I presented it, but I think is really valuable, is that the results from the thumb model, while you start off thinking everything is wrong, we've really been able to replicate the conclusions across different platforms. The original Valero Cuevas study in 2003 was performed in Mathematica. The simulations Sarah performed and were described here were implemented using combinations of Sim and MATLAB. And right now she has transferred our model back into OpenSim, and we're preparing that model for publication and release. Transporting it to OpenSim has allowed us to be able to run these simulations in a more forward way and have sort of a more realistic understanding of the experiment. So here I'm going to show a forward simulation of just activating the FPL. This is replicating the cadaveric study. You see here, we get a big joint posture change when we just pull on the FPL. If you read the cadaveric study in detail, both work from Francisco's lab and work from Joe Tulls, who was also at the VA Palo Alto, you'll see that they mention that joint posture changes when you pull on individual muscles. And based on Craig Gaylor's simulations, we know that that can have a big impact on what the endpoint force is. Of course, people were measuring everything else, and we didn't get great measurements of joint kinematics in these experiments because everything is not always possible to be measured. So here I'm showing forward simulations of activating FPL and activating the ATB. And you can see just being able to do this adds a new layer of insight to what these simulations mean and what it means for our model to be right or wrong. Sarah is actually going to be describing these for dynamic simulations and other aspects of the development of the dynamic model at the International Symposium on Computer Simulations and Biomechanics. So if you're in Brazil, please go to her presentation. So that's all I have for today. The conclusions of the talk, my basic take-home message is that in my laboratory, biomechanical modeling and simulation serve as methodological tools that advance the types of questions we can ask about upper limb function. Today, I've shown you how biomechanical simulations have helped us bridge the gaps in what we understand from very valuable experimental studies. We also have a number of studies going on where we generate new hypotheses. And in deference to the fact that we do need to understand if our model is right or understand how it fails, a critical step in my lab is always evaluation of simulation results and understanding how technical modeling choices influence results. So that's all I have. My acknowledgments, of course, are funding sources to ARMS Lab. A special thank you to Sarah Wallman for letting me focus so much on her work, and numerous people who have contributed greatly to my research.
Okay, thank you, Wendy, for a great talk. You made a very complicated topic really clear, so thank you. I also want to let you know, since you can't see or hear anyone, that I did laugh at your joke.
Okay, thank you.
Yeah. So now we'll get started with the Q&A session. As I said at the beginning, all the questions will be text-based. So find the Q&A box in your WebEx controls, and then type in your question and make sure you select to ask all panelists. So we'll go ahead and get started. I already have some questions that have come in. So first from Diogo Heralde. Apologies if I mispronounced that. He's from Imperial College. He thanks you for your talk. And his question is about the data from experimental data from cadaveric studies and what the effect might be of using cadaveric data versus live human subject data when you're validating your models.
I think that's an important question. I'm going to go back to this table from Sarah's work. I think to do something really invasive, it's typically and historically we've always taken data from cadavers. The onset of imaging has allowed us to do a lot of recent studies, and I think in particular the assessment of muscle volumes in human subjects is much improved. It's a pretty simple and obvious comparison. Muscle volumes are much larger in young, healthy adults than they are in the cadavers we have historically taken data from. So clearly, incorporating imaging data that has muscle volumes that are representative of the population you want to study is something that we consider very important. I find it interesting, though, that if you look at the results, we still could still produce bigger forces with cadaveric data than we could if we just replaced the joint kinematics. So it's interesting to note that these joint kinematics have typically been measured in cadaveric specimens as well. And so the onset of imaging and improving that data is probably something important to be considered. So overall, human subject data from imaging and in vivo is probably where we want to go, but it's really difficult to get all the information we need without including cadavers.
Okay, we have another question about the experimental data that you used. This is from Ching Sai. Did you directly measure the moment arms from the cadaveric arms that you used for the study, and if so, did they match with your optimized results? We, interestingly enough, did not do any experimental work at all in this study. It was a 100% simulation study, so I apologize if that did not come through in my presentation. What was great is that there are really talented and well-regarded and respected investigators who had provided us the experimental data we needed to get started. And given the complication of the system, rather than thinking we could go in and get better data, we used the model to explore and learn more about the data. So here's the result. This is where I got a little bit tripped up, and so maybe that's why it was confusing. Here is the result, comparing the moment arms predicted using our optimized muscle path to the moment arms measured experimentally from Kayan's lab. The author is Smuts, and the paper was published in 1998. And what we did show is that you could not accurately predict endpoint forces and moment arms of the two separate cadaveric studies. So here we're showing when we accurately predict endpoint forces from the Perlman study, we severely underestimate the moment arms from the Smuts study. And then this result illustrates that when we replicate the moment arms from the Smuts study, we severely overestimate the endpoint forces from the Perlman study. So again, our conclusion here is that while these are both really valuable studies, and we wouldn't have even gotten this far without this experimental data, they are not mechanically consistent with each other. And there's likely much more basic biomechanics work that needs to be done to clarify the mechanical actions, both in terms of endpoint force and muscle moment arms. Okay, now a question from Puya Amiri. It's more a methodological question. And her question is, what is the difference between SIM and open SIM capabilities? But to focus the question a little bit more, what parts of your work did you use SIM for versus open SIM for?
So all of the results here were completed in a combination of SIM and MATLAB. So we started with the biomechanical model from Holzbar, and here I'm just in 2005, which is a model that was developed before OpenSim existed. And this really focuses in on the kinematic representation of the thumb and the kinematic representation of the muscles and how the muscles change length with joint angle. And then we used the geometry from the SIM kinematic model and the axes of rotation and the muscle tendon path and the muscle force generating parameters, which are also a part of this kinematic model. And we transported that into MATLAB code, which was really fairly complicated, and it was started by Craig Gaylor and continued by Sarah Wallman. And basically, these equations were implemented in MATLAB. So to get from individual forces to endpoint forces, we did calculations outside of SIM. At the end of the talk, I'm showing how we have now transported that same model into OpenSim. And these videos illustrate an OpenSim model. This is a version of the Holzbar model, the Holzbar 2005 model. We have augmented it and by adding the intrinsic muscle path. And so this has all the kinematics of the SIM model, plus there are inertial parameters, and one of the things that Sarah's going to be talking about at ISCSB is the passive parameters that you need for the simulation to run. And so here you can see what we do in the SIM MATLAB model is we solve a bunch of problems computationally, and here we turn on a muscle, and the forces it generates are applied to the bones, and the dynamics are run in a forward way. So Sarah's in the process of replicating her SIM results in the OpenSim, but basically we're using the same model, and what we expect is to be able to get the same results, but to understand things like the dynamics of what happens when muscles generate forces and motion occurs. So I hope that helps clarify that question. I'm not sure it was an exact answer, but we definitely have used different platforms, and we think of the biomechanical models themselves as an anatomical library that we can use in almost any platform. And in my lab, we sort of operate over a number of different platforms, trying to pick the one that best meets the needs of the question we're asking.
Okay, so that's great. Thanks for clarifying all that. Now we have a question from Josh Anuye. Again, apologies for any mispronunciation, from the University of Virginia, working with Sylvia Blumker. Thanks for the interesting talk, they say. What was the approximate max endpoint force multiplier that was found by only altering the kinematics? In other words, how many more times the maximum endpoint force could be produced by only altering the kinematics?
When we compare these individual Monte Carlo simulation results, so this is a good example. Here, the results that we present as sort of the model that we're advocating is shown here. The experimentally based joint kinematics, the model moment arms, and the maximal muscle forces. And what Sarah's results show is that on average across all the different things that we studied, when we did 5,000 Monte Carlo simulations spanning the variability in the experimental data, we got a result in lateral pinch that was 1.2 times stronger than the human subject's data. When we replicated that same study, then the only thing we switched out was the joint kinematics, and we replicated 5,000 simulations, we had a factor of 3 weaker. So here they're both normalized to the human subject's data. So 0.4 is 3 times weaker than the 1.2. And if you look at this bigger model, you can see, you can compare, what we just did there was these two comparisons. But then you could also do experimentally based literature and imaging versus orthogonal and intersecting literature and imaging. And what you can see here is we got 5 times. So Francisco's lab's results reported the model was 4 times weaker than the experimental data. And here we showed results 3 to 5 times weaker. So we feel like we have very effectively replicated his original simulation results and verified his postulating that it's really the kinematics.
Okay, thank you. So we have time, I think, for one more question. This question is from Ashish Deshpande at the University of Texas at Austin. He thanks you for the great talk. In your study, are the moment arms analyzed for one joint at a time or multiple joints simultaneously?
The moment arm data from the literature is one joint at a time. So this is, if you go to Sarah's paper, you can see the moment arm comparisons for individual joints. But my understanding of the data is that for each degree of freedom, they did the tendon displacement method and made sure that the other joints in the thumb did not move. So the DL, d theta that they're measuring represents the change in muscle tendon length with change in joint angle for just that degree of freedom. And so the moment arms that we're comparing there are calculated the same way. So it's not clear to me what it would mean to calculate moment arm for multiple joints simultaneously, because from my own experience with measuring moment arms in the cadaver lab, we usually try to isolate just one degree of freedom moving so that all of the DL is attributable to that d theta.
Okay, I guess, but so then the moment arms that you measured at one joint were always fixed at a specific location for the other joints then?
Yeah, you know, we would have to go back to the original source data, which is the SMUTS data. And again, I'll repeat that none of the work here represents experimental work from my lab. We took advantage of the other investigators' work, which was really helpful. But my understanding of that data, there might be assessments of moment arms in multiple postures. For instance, in my elbow muscle moment arm paper, we measure elbow flexion moment arms in different forearm positions, and we measure forearm moment arms in different elbow positions. So there may be aspects like that in this data, but of course the thumb is so complicated, I can't say specifically what was done in that study.
Right, right. Okay, great.
I hope that helps.
Yeah.
So he could go to the original source data and have a better feel for exactly what was measured.
Okay, great. All right. I think we will go ahead and wrap up the Q&A session. If you could bring up my closing slides for me, Wendy.
Find my mouse right here, right?
Yeah, the next one, or next two. I think two forward. So I also want to acknowledge the funding sources for the OpenSim project. OpenSim and this webinar series are supported by several grants from the NIH and the EU, including an NIH grant that funds our National Center for Simulation and Rehabilitation Research, and we're also supported by the DARPA and the Warrior Web effort. I really want to thank everyone for a great discussion. Next slide, I think. It's really great to have questions come in from all over the world. So thank you all for participating. Information about the NCSRR, OpenSim, upcoming events, and other resources for the community are available on our website. And I'll ask that you please complete the survey that will appear in a pop-up window at the conclusion of the webinar. This will help us improve the webinar series and choose upcoming topics. So again, thank you all for participating. Thank you, Wendy, for a great talk. We hope you all continue to stay involved with the OpenSim project. Thank you.